Okay, first of all, thank you very much for this kind invitation. Going to Paris this time of year is a fantastic thing to do. And so I have a few disclosures to do. These are some disclosures I have. So I want to start with this picture, and I want to stay here for a little while. We've heard this morning from René that cancer cells, they have oncogenic signals that cause DNA damage through something we call replication stress. And so cancer cells have a higher amount of DNA damage as compared to normal cells. And this is key. This is key to also chemotherapy, how we work there. So what happens also with cancer cells having higher amount of DNA damage is that this becomes a problem when you try to replicate the DNA. So what happens there is that many of those lesions will not be repaired, and those cells will die. So in cancer, you have a lot of death. A lot of cancer cells will never grow out. And instead, you will have some clones grow out. So cancer is a clonal disease. And the reason why they survive is because they actually repair the DNA at replication fork. But what they do when they repair is that they misincorporate the wrong bases. So with incorporating the wrong bases, they generate something we call DNA mutations. These mutations are then accumulating in these cells that then go through several rounds, and cancer cells can have many mutations as compared to the non-cancer, the normal cells. And these mutations are very important in development of cancer. So this is key, this cycle, this vicious cycle of DNA repair that you have. So what we can do, and what we have done for a long time, is to overload the cells with DNA damage. So that will cause a lot of DNA damage, and most cells will go down and have no repair, and you will kill also the cancer cells. And this is what we do with chemotherapy. So we had a question from early this morning about why do chemotherapy kill cancer cells and not normal cells? Why is it tolerable? And we don't have exact answer to this. Of course, not every cell is dividing, but that is not the whole answer. So one key to this is, of course, probably the oncogenes that we heard René talk about before, that actually push the cells to drive and replicate in spite of having the DNA damage. Another one could be from Uwe about Slafen, about how normal cells could then stop cells that are damaged, while cancer cells then could just run through. We heard that Slafen is downregulated in many cancers. So all of these combined makes chemotherapy work today. But we want to do this more in the next round. And we heard about, for instance, you can have mutations in the BRCA1 or 2 genes that are involved in homologous recombination. So what happens when you get these mutations? Well, these are involved in DNA repair, so then that becomes a problem. You will have more rearrangements, but the cell can compensate for that by upregulating. So these VC8 cells that lack BRCA2, they have higher level of this PARP activity. You can see this PARP activity here. So they have higher level of PARP activity, and they are needing PARP for survival. So if you use PARP inhibitors, you can kill off these cells. And this has then been translated into the phase 3 trials, and we see this fantastic response. And it sounds like such easy thing, and this was a home run. It was not. It was by very hard work from dedicated scientists like Sophie, clinicians actually. It is the clinicians that made the difference here. So we got approval in 2014. But what happened was that these were the first sort of response rates that we saw in the first phase 2 trials. And here you can see that you get some partial response. In this study, they didn't have a single complete response. And you can see that also not all the responders have BRCA1 mutations. Many are non-BRCA mutations. And they are still sensitive. And now we know that there are other mutations, there's other way of getting rid of BRCA1 and 2 other than just standard mutations. So at this time point, they didn't call the mutations so accurately either. And then going back and seeing how did the response rate look like from this trial. And then you can see that the patients had a progression-free survival about 6 months. And their BRCA status didn't matter. So at this point, AstraZeneca, who was running this trial, decided to close the program. And one reason why they got these results is because we are working with end-stage patients. So one very important, and they were multi-resistant to platinum and other drugs that also gives resistance to PARP inhibitors. So working up the ladder and adding on to PARP inhibitors in first-line therapy, as I showed you in the first graph, makes the difference. But that was not all the difficulty. Another difficulty that we had in the beginning was that different inhibitors were differently efficient. So all of these inhibitors, PARP inhibitors, are potent, low nanomolar inhibitors of PARP. But some doesn't kill BRCA-defective cancers, some do, and some are really efficient. So clearly, this makes no sense. If you inhibit PARP, why don't you kill the cells? So this was actually sort of highlighted by Hugues de Thé to demonstrate that there is differential trapping. Previously, PARP trapping had been described, but it had never been described that compounds could differentially trap it. It was thought to be related to the inhibition of the catalytic activity. So here, the trapping really solved that. And this was much later. This Junko's paper was in 2012 in Cancer Research, and I should probably refer to that one, that paper. And so there are many things going on. We have PARP involved in this single-strand break repair, which has been originally described. We have PARP trapping, and PARP is also involved in fork protection and restart. But further to that, and there's different effects with inhibitors on these, but also PARP is involved in backup end joining, micromediated end joining, and a whole lot of other things. And there's now more than 5,000 papers describing the PARP and BRCA sort of synthetic lethality. And there is a new sort of way of describing this every time I open an issue of Nature or so. So it is not simple. It is complex. But that should not stop us in the clinic. We have to progress. And this also stressed the importance of understanding the molecular mechanism that we have. So how do we go on and find new targets? So we've talked about a lot of the DNA damage that you can have endogenously from oncogenes or from chemotherapy. And this is recognized by these kinases that you've heard about today that actually recognize the damage and then signal it. And they signal to DNA repair that then completes the repair. And we've been trying to target many of these. The PARP inhibitors was very successful because of this huge synthetic lethality, this huge difference of killing the mutated cells, the BRCA mutated cells, and the non-mutated cells. The other ones have been struggling a little more. We heard about WEE1 and CHEK1 inhibitors this morning, and also ATR inhibitors from Hugues de Thé. They have been struggling a little harder in the clinic because there you don't have the same therapeutic index. So we were thinking, how are we going to select a new target? So actually, in order to complete DNA repair, you need building blocks. So here you lack building blocks. Here you need building blocks, nucleotides. And nucleotides in cancer are generated through a different pathway than in normal cells, because there is cancer-specific metabolism that contribute to the generation of nucleotides in cancer. So one of the proteins, MTHFR2, we were interested in this one. And we were interested in this one because it came out of a screen of 22,000 proteins. And the top protein actually, the that was an enzyme was MTHFR2. That is the most overexpressed enzyme in cancer, looking globally at cancer. So we were interested in this one, but funny enough, we were not the only one interested in this one, because it was published in this obscure journal. People saw it, and, and everybody jumped on it. And they could use RNAi to validate this and say that if you remove this enzyme, cancer cells die. And we could do the same. If we remove the enzymes, cancer cells die, and then you can express the wild type but not catalytic dead. And then you can rescue the survival, but not here. So this we thought, okay, makes it a good target. So, so how does it work? Well, MTHFR2 is contributing in the one-carbon metabolism. So it brings a carbon to purines or to pyrimidines and to generate thymidine, nucleosides, hypoxanthine, and so forth. And we could see that we could actually rescue the loss of survival just adding in thymidine. Thymidine is used to generate DNA. So we love this because we like DNA replication, which I mentioned in the beginning. So the idea is that MTHFR2 generates this methyl tetrahydrofolate, and then add this methyl group. It takes this methyl group and then put it onto the uracil. This is what thymidylate synthase is doing to generate thymidine, which goes into the DNA. So, so if we inhibit this enzyme and we see that it's rescued with thymidine, maybe this is the mechanism of action. So removing MTHFR2 with siRNA, we could see reduced fork speeds. So in this assay, it's called the DNA fiber assay. So we label the DNA with chlorodoxyuridine, iodine deoxyuridine, and then we stretch the DNA out and see how fast the DNA replication is going. And you can see that it's going slower, replication stress, when you remove MTHFR2. And you get this gamma H2X DNA damage and these RPA foci, which is what we generally call replication stress. So this sounds great. This is what we are working on. This is a perfect target. This is what we want to do. So, and we can see it localizing, even the DNA damage is localizing to the sites of DNA replication. So this is perfect. So let's make inhibitors. And we purified the enzyme, which was very, very difficult. And then we set up a very expensive assay. And then we screened through 11,000 compounds and got no hits. So what do you do then? They came to me and asked, we want to screen more. And I said, well, it's going to be expensive, but they want to screen more. Okay. We screened 43,000 compounds and no hits. So, and then they said, we want to screen more. And I said, well, we can't really afford it, but European Union can. So there's a European lead factory, which is set up as a joint initiative in Europe, located in Belgium, which then can screen 500,000 compounds. And we went to them. It took them 2 years. It was very expensive. And they came back with us with no hits. Now, European lead factory has unfortunately closed. Because, I guess, for EU spending and things like that. Anyhow, so what do you do? As an academic, you typically, drug companies probably would give up, but as academics, you don't. So we solved the crystal structure and published this crystal structure. And to help to make the crystal structure, we actually found a compound that was from Lilly that could bind to the MTHFR2 protein. So that helped us to generate the crystal structure. And then instead of doing the screening, we started from that compound and as a hit molecule, and then did hardcore medicinal chemistry. This is actually the work of 15 chemists over a period of 3 years. And then we generated a really potent inhibitors that was low nanomolar inhibitors that killed cells also at low nanomolar concentrations. And they looked like this, these compounds. So, so when we use these compounds and see, the first thing people ask is, do they hit the target inside the cell? And the target is MTHFR2. So if we, normally you can see it on Western blot at 37 degrees, it's soluble. But if you increase the temperature to 57 degrees, then now it's denatured, like if you boil eggs. The egg protein is turning white. So it's the nature, and it cannot go out in the Western blot. If you try a boiled egg and run it through your Western, it doesn't work. But if you then titrate in your compound at a higher concentration now, the compound stabilize the protein. So now you can start to see that it's soluble. So now you can calculate at what concentration the inhibitor binds to the target inside the cell. And so we've done this with different compounds. This 9078, 28, is the most potent one, but it didn't stabilize as well as this 9619. And the reason is because this can be polyglutamated. When entering into the cell, it can be polyglutamated to prevent it to leave the cell. So it's taken up much more efficiently. And then you can see that and these cells are non-cancer cells. So these are cancer cells, but these are non-cancer cells, and they have less uptake of this compound. And you can see that now it is not stabilized in non-cancer cells to the same degree. So this polyglutamation, which is basically this part of the molecule, which is polyglutamated, is a characteristic that exists on methotrexate and pemetrexed, which are chemotherapy drugs. And this is probably one way for these methotrexate and pemetrexed to be used in the clinic, because it is taken up in cancer cells much more efficiently than it does into normal cells. And it's polyglutamated in there. So we also keep this feature to our compound, which is very beneficial. So then the question is, now we've seen that it goes and binds in the cell. Do they work as we expect them to do? Compounds typically never do, but these compounds actually did. So you can see that now, with the inhibitors, we slow down with the replication forks, and we get this gamma H2X specifically in S-phase cells. We get this replication stress. This is great. So, and then you make these kind of models on that you think how they work, the compounds, and those models are typically always incorrect. So to test if this is correct, this model, we added in thymidine. And if we add in thymidine, if it's going through this pathway, we should reverse all the effects. And what we see when we add in thymidine is now is the fork speeds are up again, and we rescue survival to a large extent. So this is very nice. We see that actually it seems like this model holds up. So if this holds up, this model, then if we inhibit this enzyme, DUTPase, and it has been developed DUTPase inhibitors before. If you look at DepMap, DUTPase is the best anti-cancer target of them all. So if you lose this enzyme, all cells die. So many people have made inhibitors, but the inhibitors do nothing. So it doesn't always correlate with losing the protein and inhibiting the protein. But these DUTPase inhibitors have been tried in the clinic together with thymidylate synthase inhibitors, 5-fluorouracil, and they are very toxic. But together with our compound, maybe we could, if we remove this substrate and that substrate, with combining the two, they should synergize, at least on paper. So we can see when we combine them that now they synergize like mad. And if we then add in thymidine, we can reverse this completely. So it works very, very nicely. And you can see here that when looking then into the uracil being incorporated into the DNA, we can see that in the combination, if we inhibit both of these, we get a lot of uracil incorporated into the DNA. And that kind of asks the question, how are these compounds toxic? Is it because of loss of thymidine, or is it because of incorporation of uracil into the DNA? So we could test that by looking at the protein involved in repair of uracil in the DNA, which is SMUG1. So if we knock out SMUG1, we can see that these become even more potent. And so indeed, the incorporation of uracil into the DNA is very important for the mechanism of action. So the reason that we picked this target in the first place was that it was a cancer-specific protein. We can see that our inhibitors slow down replication fork and cause this replication stress. But this is also done with methotrexate or ATR inhibitors or any other chemotherapy drug that we have. The difference is that we don't see this in non-transformed cells. So here, we don't slow down replication forks, and it's not toxic to proliferating 3D4 positive bone marrow cells. It's only toxic to the cancer cells. And then we can grew this in mice, and we can see that our effect is more efficient than cytarabine, which is nice. But then the question is, are we hitting the target in here? And to test that, is this actually tumors suppressed by us actually reaching the target? So we took out targets, and again, we started to boil these cancers. Rather than boiling egg, we boiled tumors. And while boiling this tumor, we can see that the protein is more soluble. And so you can see that you have target engagement in these tumors, which is very nice, of course. But then, of course, the other question is the compound selective? Normally, you have compounds that do a lot of different things. So the first thing to do is, of course, to test all the other compounds that also bind tetrahydrofolate. And we can see that thymidylate synthase, SHMT1, DHFR, MTH1, DHFR activity, and so forth. None of these are binding or inhibited by the compounds, as compared to pemetrexed or methotrexate, which are broadly acting, inhibiting these essential enzymes. And then we can go through kinase panels, and it's clean, or safety panels, and it's clean, and that looks very nice. But then we come into a problem. And the problem is that the protein, we're targeting this DC domain of MTHFR2. But this DC domain also sits in D2L and D1 and D1L. This one is not so important. These two are not so interesting. But the D1 is very interesting. So we're hitting this one. So we also hit, because they are structurally, we made the crystal structure for these ones. And we see that they're almost identical, and we see that the compounds inhibit all of them. So we don't have selective inhibitors as compared to the isoenzymes. So then the question is, what do we target? Is it the MTHFR2 that is important, or is it MTHFR1 or the other ones? So if you go to the Bible and the textbook, you should see if you remove the target, the compounds should not be toxic anymore. So if we move D1, you can see the compounds are still fairly toxic. There's a little difference. But if you remove D2, then they become resistant at higher doses. So it seems like D2 should be the target according to the textbook. But then we know, and now I've showed you everything. It's so easy so far, but actually this is all wrong. So now I'll give you the real story, how it works. So MTHFR2 is located in the mitochondria. And what it does there is that it releases formate. And this formate is then made into 10-formyl tetrahydrofolate, which then feeds in and gives both thymidine and purines. But most importantly is the generation of purines. But thymidine is not a purine. It's a pyrimidine. So it's a bit strange that our inhibitors was affecting this pathway and not that pathway. And that is also potentially because we inhibit this pathway here. But then, so we didn't really understand this. But the first thing of it is that, formate release should be affected by our compound. And if we take MTHFR2 knockout cells and look at formate release, we have no formate release. But if we look at our inhibitors, we have a lot of formate release, even higher with the inhibitor. So if the inhibitor would inhibit MTHFR2, the activity should not ideally go up. It should go down. So this is somehow not correct. And then, the data is correct, obviously. But our interpretation is probably not correct. And sodium formate didn't rescue like thymidine did. So there's something going on here. And what we realized is that this 9619 is structurally very similar to methotrexate and pemetrexed. And those do not enter into the mitochondria because there is a specific transporter into the mitochondria. So we then tested and isolated mitochondrial DNA and saw that the MTHFR2 wasn't stabilized in the mitochondria, only in the nucleus. So actually what happens is that our compound doesn't go in and inhibit MTHFR2. in the mitochondria at all. Okay, so what is happening then? So basically, this is how our compound works. It inhibits the DC domain of MTHFR1 here. But if this is the case, then if it inhibits this one, you would generate purines from there and pyrimidines from there, and it shouldn't do anything. So, so it shouldn't work, but it works. So then we played around a little bit and dialyzed the serum. So if we use dialyzed serum, which doesn't contain nucleotides, then now it becomes less efficient. And then we thought, so why could that be? And that could be because in humans, there's hypoxanthine. And if we dialyze the serum, you remove hypoxanthine. So hypoxanthine, if we add in hypoxanthine, we could resensitize these. And hypoxanthine actually blocks the de novo purine synthesis pathway in a feedback mechanism. And then, if you have formate release, you could generate this formate trap that wouldn't then generate thymidine to produce thymidine. So this is more of a complex mechanism that we generate a formate trap with our inhibitor, that it depends on MTHFR2 activity. So if this is correct, adding in hypoxanthine, then if we then add in thymidine again, we should get a rescue, and we get a complete rescue with thymidine and hypoxanthine. So let's go to MTHFR2 knockout cells. So in the knockout cells, they're actually alive, which is strange, because a lot of people validated it with siRNA and say that the cells are dead. But in that map, they should be alive, and if we knock it out, they also become alive because they use this pathway as a backup pathway. They activate this pathway. So, this pathway is there. If you don't have MTHFR2, now, if you treat with our inhibitor, one would expect that we wouldn't get purines, because now we have no way of generating purines. And so here, they are very sensitive, actually, to our inhibitor in dialyzed serum. So if we add in hypoxanthine, now they are rescued. And this is what we saw before, that there is no toxicity when you have normal media which contains hypoxanthine. So, so indeed, this seems to be the mechanism. And then if we would add in sodium formate, that would also rescue. So now we can also see it's all about the purines here. And, but then the chemist who has been involved in this project and really been eating and living and sleeping this one, his name is Martin Henriksson, he thought, well, what if we add in both? Then we would artificially recreate the trap. So hypoxanthine rescues, sodium formate rescues, but adding in both, now you resensitize them again because you artificially generate this trap. And then that would then make them, rather than not be able to generate purines, now they wouldn't be able to generate thymidine. So this should be rescued with adding in hypoxanthine, sodium formate, and adding in thymidine should rescue it. And that's exactly what we see. So this is a complex way of identifying the detailed molecular pathway. And you can see that the difference here is, here we kill off cells at low nanomolar concentrations, and here we go up to, you know, high micromolar. So this is truly the mechanism of action. At least we hope so. And similarly then, if we reactivate the de novo synthesis pathway by adding in ICA, we should also be able to rescue, and we do. And another way of seeing this is from the CRISPR-Cas9 screen that we did. The first top hit that we found was the SLC19A1, which is the folate transporter. So this is what is transporting the compound into the cell. The next one is HPRT. HPRT is converting hypoxanthine into IMP, and that blocks the de novo synthesis pathway. So mutating this one, now you don't get inhibition of the purine pathway. You release this folate trap and generate tetrahydrofolate, which then can be used to make thymidine. So this all makes sense. So everything kind of adds up, and that makes you very happy that now all of a sudden it works. So how our compounds work is that they generate this folate trap, which is actually dependent on MTHFR2 activity. So when we do the CRISPR knockout and get rid of MTHFR2, you don't get formate release and you never generate the folate trap. So this is the reason why they become resistant. But that wasn't because they hit this target, because it's dependent on that target to generate the toxic product, which is the folate trap. And so this then takes us into, how are we translating things into the clinic? We use mice models. Mice have high level of folate, high level of thymidine, and low level of hypoxanthine, which is opposite to humans. They have low level of folate, low level of thymidine, and high level of hypoxanthine. So our compound should actually not work very well in mice. It should only work in humans. And we're not the first to hit this pathway. 5-fluorouracil, for instance, and many of the other pemetrexed and things like that, they don't work in mice models. They work in humans, and they are first-line therapy since 5-fluorouracil since 1957, and it hasn't been replaced. But if you try it in a mouse model, it doesn't work. So maybe mouse models, maybe it's something wrong about the model. Yes, there's something wrong about the model. So, so if we want to but then if we want to develop treatments, people say if it doesn't work in mice, it's not going to fly. So we have to make it work in mice. And in order to do that, we actually knock out thymidylate kinase. And the tumors grow and they are fine. But now they cannot use thymidine to rescue. And now we can see very good effects also in mice, really good responses. And talking about how the cross-reactivity to immuno checkpoints, we see PD-L1 expression go up after our treatment in a similar way with cisplatin. So potentially this could also be used to increase the immune response. So what makes me kind of excited about this? Well, we tried the PARP inhibitors, and if you see, the PARP inhibitors had a huge therapeutic index. It's a 200-fold therapeutic index, and that made the difference in the clinic. Now, ATR inhibitors are quite hyped in ATM knockout cells. And this is not true for all cells, but in some cells, you get as much as 6-fold difference in sensitivity to ATR inhibitors. And that could be a nice therapeutic index. But here, if you look at these colorectal cancer cell lines that are KRAS mutated, they are sensitive at low nanomolar concentrations, while the other cells are completely resistant. And this is actually true for many of some cancer cells are completely resistant to our compound, while others are then sensitive in the low nanomolar range. And it seems very black and white. And we have failed we kind of hope to find a biomarker, but we are not there yet. We need help. We need more scientists to actually figure out what is the biomarker for this one. Because it's not going to work for all cancer. It's going to work for some cancer. And as always, MTHFR2 has other roles. It doesn't only work in mitochondria, it also works in the nucleus. So after DNA damage, ionizing radiation, MTHFR2 is relocating very, very rapidly into the nucleus. And this relocation is dependent on ATM and DNA PK. So these proteins activate a nuclear localization of MTHFR2. And what it does there is that normally after ionizing radiation, you form this RPA foci. And we have heard about them before. These are coating the single-stranded DNA. And in the absence of MTHFR2, that's not the case. So also we heard about RAD51, which is required for homologous recombination. You can see that when you remove MTHFR2, you don't form these RAD51 foci. And you can see that if you remove MTHFR2, you don't catalyze homologous recombination. So now I've been talking for a while, but if you remember in the beginning of my talk, I mentioned that if you have a recombination defect, you can combine that with a PARP inhibitor and kill off those cells through synthetic lethality. But there is also a lot of cancers that are not recombination defective, or those that have reverted to become recombination defective. And most cancers are BRCA wild type. But if you would use these inhibitors, which then now targets MTHFR2, which is a cancer-specific protein, this cancer-specific protein would make the cancer cells defective in homologous recombination, and then together with a PARP inhibitor, you would get induced synthetic lethality. That would be specific for cancer cells. So if you look in non-cancer cells, either olaparib or a MTHFR2 inhibitor in this case, they do nothing. But combined, now in cancer cells, they are very efficient. So we think that this could be potentially a pathway to go forward. So now I've talked to you about that synthetic lethality, and it's actually very difficult. The mechanism of action is difficult. The translation into the clinic is difficult. And then I told you about the MTHFR2, which is the most overexpressed proteins. We made inhibitors to MTHFR2. It turned out that we inhibited MTHFR1 instead, but that was equally good. And we get this cancer-specific replication stress by thymidine loss. We create this folate trap, which is then mediated through MTHFR2 activity. So the CRISPR-Cas9 actually is very poor in predicting drug targets. People are using this as the Bible. PARP is not coming out there. DUTPase that I showed you is the top target, which is not a good target. MTHFR2 is not a good target according to this. So it is not necessarily the best thing to do. And I also told you a new role of MTHFR2 in the DNA damage response and modulating immuno-oncology drugs. Okay, thank you very much. And this is the team, yes. This is the team and the people involved in this. Thank you. Yes.